So, yeah, thanks, Zoe, for the kind introduction. Hey, everyone. So, I'm Stefanie. I'm a PhD student at the Department of Statistics at LMU Munich in the working group of Christian Müller, which is called Biomedical Statistics and Data Science. So, first of all, thanks a lot for having me and for giving me the opportunity to present my work here in this seminar series. So, let's talk about microbiome networks. So microbes are all around us. And besides common analysis methods like abundance-based methods or a PCA or maybe differential abundance analysis, microbial association networks have gained increasing interest during the last decades. And this is what we're going to be talking about today. Since some of you might not be familiar with these kind of networks, I want to start with a short introduction about microbiome networks. So these networks consist of nodes or vertices, which are the microbes, in our case, like ASVs, or in older data sets, you might find OTUs, or these can also be taxa on a certain level, like genera or families. And they are connected by so-called edges, which represent the microbial associations. So you can imagine these as correlations, for instance. And there's also an edge color here in this plot, and they just represent the direction. So positive correlations or associations are colored in green, and red ones are represent negative associations. And these single microbial networks give you an answer to questions like how are certain microbes associated, or what's the structure of the microbial community or of certain subcommunities. But researchers might also be interested in different groups. So let's say you have two groups in your data set, like cases or controls, or like different environmental states. Then you might ask if the networks change across these two conditions. You might want to know if certain associations change between these two conditions, or if the overall network structure or certain network properties change. And this is what I have written my R package NetCoMi for in the course of my PhD studies and with the great support of my supervisors. And meanwhile, NetCoMi is a comprehensive R package for constructing, analyzing, and comparing microbiome networks. The package is available on GitHub, and there's also a comprehensive documentation, which you can find at netcomi.de. And we also published a corresponding paper a few years ago. There you find the methodological background about NetCoMi, and also the package itself is described in this paper. So let's have a look at the NetCoMi workflow. So NetCoMi has a modular structure with different functions. There's one function for network construction. This is actually the most important part. Then there's a part for network analysis. So you could also use NetCoMi if you already have your association matrix at hand, and you could start at this point. You can do network comparison, as mentioned before, and there's functionality for constructing differential networks. So in these networks, two nodes are only connected if they are differentially associated between two groups. And I want to briefly give you an overview of these different steps. So let's first of all have a look at the network construction step. Such a read count table here on the top left is what you usually start with. So here you have samples in rows, your ASVs and columns. This can also be like families or genera. And the cells correspond to read counts. And here on the right-hand side, we have an association network, what we usually want to end up with. And this network is represented by a so-called adjacency matrix, where each entry represents to an edge in the network. And this, in turn, is a sparse version of the association matrix, which we have to compute. So this step here, from read counts to associations, is critical because we have to take the typical characteristics of microbiome data into account. Like for instance, the sample we observe is only a sample of the true underlying microbial composition. The data are typically compositional, meaning they carry only relative information. The sequencing depth varies usually across the samples. The data are highly zero-inflated, meaning you see a lot of data, of zeros in your data. Like here, for instance, there are only a few, and you usually have a few dominant taxa, like here, this one, for instance. And the data is usually high-dimensional, meaning the number of taxa or microbes is much larger than your observed sample size. Now, let's have a closer look at this step here. First of all, you need a proper zero replacement. In a simple case, this could be just adding pseudocounts to your data, but there are also more sophisticated methods. Then there's usually a normalization part or transformation to account for the compositionality aspect. A well-known method is the CLR or standard log ratio transformation, but there's also other methods available. And then the most important step is association estimation. Here you could simply use correlations, but we prefer using conditional dependence-based measures because we are usually interested in the direct correlations between each two taxa conditioned on all other ones in the network. And as you can see, there's a bunch of methods out there. For most of them, there's also corresponding R packages. And our intention when starting with the NetCoMi project was to provide one single R package where you have all these methods at hand and only need to adjust a few arguments to have access to all of these methods. Okay, so let's assume you have now constructed your network. Then the next step is network analysis. Here's a short overview of the methods provided by NetCoMi. And here in the middle of the slide, you can always see some highlights in the network plot, which is also done with NetCoMi. So you have the option to highlight some centrality or highlight centrality measures by node sizes, for instance, here. So larger nodes mean that these taxa are more important in the network. You can use common clustering methods and use them or highlight them by different colors in the network plot. There's also an option to compute hub nodes, and they can be also highlighted here by like bold edges. And yeah, these hubs are usually the most important nodes in a network. And there's also a bunch of global network properties. These are not included in the plot, but there's also a summary which you can just print. Okay, and as mentioned before, there's also the possibility to compare two networks between two groups. Here, we want to look at an exemplary network comparison for a subset of the American gut data. So this is taken from the Speakeasy package. And yeah, the American gut data is a large collection of 16 sRNA sequencing data. And I used these data on genus level. And for this example, I just wanted to compare subjects with seasonal allergies versus subjects without seasonal allergies. And here you can see a simplified R script. And this is in principle everything you need to type in your or type in SR code for this whole approach of network construction, network analysis, then producing a network plot and network comparison. And of course, for each of these functions, there's a lot of arguments which you can adjust to your needs, but in principle, it's only a few lines of code. And here's an exemplary network plot where we compare the networks between these two groups, seasonal allergies versus no seasonal allergies. And as you might see, the same layout is used in both groups, which is usually done with NetCoMi so that you are able to compare certain associations at first glance. So for instance, you can see that this association here is high in both groups, whereas other associations like this here is existent in one group, but not in the other one. You're also able to see hub nodes at a glance and so on. And here, instead of clusterings, the node colors represent the phylum level. And here's a part of the summary output of this network comparison. And yeah, you can see that for all these network measures, you get the values for both groups and also the difference. And if you performed permutation tests, then you would also get a p-value. So testing is also included in this package. Yeah, so if you're interested in using NetCoMi, feel free to visit the website. There's also a great online book called Orchestrating Microbiome Analysis, which we contributed to with 2 chapters. And here are my contact details. So feel free to get in touch if you have questions. And yeah, then thanks for your attention.